If I can figure it out, you too can figure it out. With this motive, let's begin the lecture on immunity. Simply, the immunity. What is immunity? One line definition: immunity is the defensive power of the body. Okay, I wrote defensive power of body. Obviously it is against illness, any of the microorganism, any of the antigens. So that is immunity. Now proceeding forward, the thing that we need to know about in the topic of immunity is we need to know about its type. There are two types of immunity. One is innate immunity and another is adaptive immunity. Okay, we need to know the basic difference between one, and I'm going to mention it like this. Okay, now innate immunity from word innate, it is itself clear that innate immunity is the immunity that is present by birth. The word innate means by birth. So I'm going to write here one thing, and the thing is present from birth. Okay, since it is present from the birth, it is also given other two names, and that is natural immunity and native immunity. And it is also called native immunity and natural immunity. Okay, now since it is present before the birth, that means we are born with this immunity, native immunity or innate immunity. It is also called the first line defense. So I'm going to mention here first line of defense. Okay, and other three terms that I'm going to mention here are: it is non-specific, it is non-specific in the sense that whatever antigen or whatever microorganism attacks our body or attacks human body, at that time it is not specific for any of the antigen. It will act to each of the antigen in similar way. So it is not specific. We are saying it is not specific because we have got here another type, that is adaptive immunity. It is specific. It is specific because the adaptive immunity is produced whenever antigen enters in our body or whenever microorganisms enters in our body. So it is specific to particular microorganism or for particular antigen. So it is non-specific, we have mentioned here. And another thing is it is or it has, okay, let me write, it has no memory. It has no memory. Okay, I'm going to elaborate this term when we'll be writing points on adaptive immunity. So hold it, don't worry. It has no memory. I mentioned it has no memory. And another thing that we are going to write here is there should not be prior antigen exposure to body to produce innate immunity. This is also the same case of above one. I mean, we are mentioning it here because in adaptive immunity, to develop adaptive immunity, there should be exposure of antigen. Okay, let me write here: no prior exposure to antigen. Okay, now these are the points on innate immunity. Now let's move toward adaptive immunity. And under the topic of adaptive immunity, since it is not present during the birth time, it is acquired through the lifetime. I mean, we are acquiring in our lifetime. It is not present in our birth time. So it is also called acquired immunity. Okay, we said that it is acquired in our lifetime. How it is acquired? The answer of question how it is acquired is we acquire it whenever any of the antigen or any of the microorganism enters in our body. That means we acquire it whenever antigen exposed or our body is exposed to antigen. So we can say here, as we have written here, no prior exposure to antigen in the case of innate immunity. But in the case of adaptive immunity, we have to write here prior exposure is present. Prior exposure of antigen. Prior exposure of antigen is present. Okay, that means suppose we can suppose it is my body, it is my human body, and it is my body, and for the first time one microorganism is trying to enter in my body, and it is successful, it has become successful to enter in my body. So I'm focusing on the point for the first time. For the first time, any of the antigen or microorganism is trying to enter in my body, and it successfully got entered in my body. For the first time, my body will take a bit time. It will take a bit time to recognize that antigen, and it will take long time to recognize that antigen, and as it recognizes that antigen, it will produce specific antibody against that antigen. So we wrote here prior exposure of antigen is present, because to develop adaptive immunity, to develop antibody against that particular antigen, we need to get ourselves exposed to antigen. So we wrote here prior exposure of antigen is present. And another thing that we have mentioned here is, we mentioned here it is non-specific, but in the case of adaptive immunity it is specific. We wrote here it is specific because we said the antibody produced will depend upon the antigen that is exposed in our body. And another thing that I was mentioning is the adaptive immunity has memory. What does memory means? As I already gave example earlier, whenever the antigen tries to enter in our body for the first time, our defensive mechanism of our body will take a longer duration to identify that antigen. But once it has already been exposed with that antigen and has produced antibody against that antigen, for the second time, if same antigen will attack our body, then at that time there will be no that much long duration in creating antibody, because it has already kept the identification of that antigen in its memory. So we say adaptive immunity has memory. Okay, I think you are clear. Memory, it has already kept in its mind, let's not say mind, we can say that in its memory, that this antigen, if this antigen will come and attack me, then I will produce this antibody. So let's say it has memory. Okay, I think I made clear. And as we have already mentioned here that it was the first line of defense, similarly, if it was the first line of defense, then obviously it is the second line of defense. Okay, we have made the few points on innate immunity and adaptive immunity, and going further we can see that innate immunity, these are the component of innate immunity. Let's not go toward the component right now. I want to mention the timeline here. When specific antigen tries to invade our body or tries to get into our body, at that time innate immunity will come into action for about 15 hours of initial infection. But after 12 to 15 hours, then comes the role of adaptive immunity. That's why it was called the first line of defense. First line of defense. And this was called second line of defense. Okay, now let's move toward the component of innate immunity. What I'm writing here is component of innate immunity. We have previously mentioned that innate immunity is the immunity that is present in body before the birth. So component, let's talk about component. And while talking about component, the first thing that we need to talk about is barriers. barriers, barriers. Now what are these barriers? Barriers, obviously the blockers that prevents. Okay, under barrier we can take, there are two subtype, and the two are, one is anatomical barrier, and another is physiological barrier. And talking about anatomical barrier, we have example of intact skin. Obviously our skin is has protective action. It helps to prevent microorganism to enter in our body. So intact skin is the example of anatomical barrier. And what about physiological barrier? Talking about physiological barrier, we have secretions. What I said is secretions, such as our saliva contains what? Lysozymes. Okay, similarly, and these lysosomes helps to fight against bacteria, various microorganisms. So it is the physiological barrier. Similarly, our stomach. What our stomach contains? It contains HCl, and HCl have similar activities. It fights against infection also, microorganism also. And these were the example of barriers. Now talking about cells. I mean the component. We are under the topic of component of innate immunity, and under the component of innate immunity we completed barriers. Now let's talk about cells. And talking about cells, we got here phagocytic cell. Phagocytic cells. What are these phagocytic cells? The meaning phagocytic cells means the cells that engulfs microorganism or antigen that tries to interact in our body. That is the example of phagocytic cells. So what comes under phagocytic cells? Neutrophils and macrophages. Neutrophils and macrophages. Okay, neutrophils and macrophages. Now these are the phagocytic cells, and there are other cells that we need to mention, and among the other cells we have natural killer cells. Natural killer cells. And we have dendritic cell. Don't worry, we are just taking out, we are just telling name right now. Later on we'll explain each and every one in detail. Dendritic cells. These are the example of cells that are the part of innate immunity. Similarly, talking about another component, and the name of another component is plasma proteins. Plasma proteins. Okay, on the plasma proteins we got the component such as, we can say, C-reactive proteins. C-reactive proteins. Similarly, we have protein of complement system. Protein of complement system. Okay, protein of complement system. Similarly, we have other things also. Mannose binding lactate. Mannose binding lactains. Okay, these are the example of what? Component of innate immunity. Okay, these are the example of innate immunity. Now we need to focus on one thing, and the thing is how, I mean what these cells or what these talking about what these cells contain in them that makes them to function or to perform the action for innate immunity. Okay, what I mean to say is there is particular receptor. There are particular receptor that recognizes particular molecular pattern in pathogen. What I mean to say is, okay, let me clear. The innate immunity is due to the presence of, I mean, what makes the innate immunity the innate immunity? Innate immunity is due to the presence of receptor that recognizes certain pattern. That means pattern recognition receptor. This pattern recognition receptor are present in leukocytes or WBC. Present in WBC. Okay, these are present in WBC, and WBC is a part of innate immunity. Okay, this pattern recognition receptor receives or recognizes PAMPs. What does PAMPs means? These WBC, these receptors which are present in WBC, they recognize as PAMP. And what are these PAMPs? These PAMPs are pathogen associated molecular pattern. Pathogen associated molecular pattern. Okay, you need to just focus on words. Pathogen associated molecular pattern. The molecular patterns which are associated with pathogen, that means the microorganism or the antigen, they contain PAMPs, and those PAMPs which enter in human body are recognized by pattern recognition receptor which are present in WBC. Now I think you got clear about these things. Okay, now while talking about PAMPs, we need to talk about DAMPs also, and what this DAMPs means. This DAMPs means damage associated molecular pattern. Damage associated molecular pattern, same as EVOC. And where are they present? They are present on infected cells or the cells which are harmed by microorganism antigens. And why? And these are also recognized by whom? These pattern recognition receptor, and they are also removed from the body, and that is essential to remove them from the body. And this is how we talked about pathogen associated molecular pattern and DAMPs and pattern recognition receptor. What causes innate immunity? Innate immunity, presence of pattern recognition receptor on WBC that recognizes PAMP and DAMPs, and that's all. Okay, we're talking about these things. Now we would like to move in detail about, as a whole we said that there is presence of pattern recognition receptor in leukocytes. Now we need to talk about specific receptors. Now what are these specific receptors? Okay, let's put a topic: pattern recognition receptors. Okay, under pattern recognition receptor we need to learn about what we need to learn about. We need to learn about the specific receptor. Okay, such as this is cell. Okay, let me draw it. This is cell. This is cell. This is cell. There are several receptors. Some are present. There are several receptors. Some are present on the surface, whereas some are present inside the cell. And the one that are present inside the cell, they are called cytosolic. What I said? Cytosolic receptors. Or cytoplasmic receptor. It's obviously simple. Inside cell there is presence of cytoplasm, so we are telling it cytosolic or cytoplasmic receptor. And outside the cell, the receptor that are present are called to be peripheral receptor or plasma membrane receptor. So outside the cell, the receptor that are present are called plasma membrane receptor. Okay, plasma membrane receptor. Okay, now while talking about plasma membrane receptor, we have got one important receptor, and that is, and we're talking about that receptor. The receptor is, okay, toll-like receptor. What I said? Toll-like receptor. And another receptor that is present on surface, and that is C-type receptor. Let me draw here. This is C-type receptor. And we wrote the example of plasma membrane receptors, toll-like receptor and C-type receptor. And while mentioning our cytoplasmic, cytosolic or cytoplasmic receptor, we need to mention about what? We need to mention about node-like receptor, node-like receptor, and another one is rig-like receptor. Okay, now these two are cytosolic receptor, whereas the above two are plasma membrane receptor. Now toll-like receptor. Okay, you got the idea. Okay, I'm gonna repeat it again. There is microorganism or antigen in outside this cell. This antigen or microorganism has what? PAMP, pathogen associated molecular pattern, and this PAMP is recognized by whom? This toll-like receptor here, because it is what? It is pattern recognition receptor. It will recognize this PAMP, and after recognizing this PAMP, what will happen? The thing that's gonna happen after it recognizes it is it will be activated and will produce substance. The substance like interferon gamma, and another nuclear factor K beta. Okay, microorganism or antigen came. Microorganism came. It binded to this toll-like receptor. Toll-like receptor became active, and it's gonna secrete NFKB or nucleus factor K beta and interferon gamma. Okay, it is going to secrete these things. Now what? Now what will happen? These are secreted. Now what? Okay, when NFKB or nucleus factor nucleus factor K beta is produced, then it is responsible, whenever this will be produced, it will be responsible for recruiting more WBC at the site of infection. That means more amount of WBC, it will call more amount of WBC here to fight against these microorganism which has here, or let's call it antigen which was here. So more amount of WBC will be recruited at the site of infection, okay, this was it. And what this interferon gamma does? Interferon gamma, obviously interferon, it is what? Antiviral. It will fight against viruses if they are present there. So this was all about toll-like receptor. And what C-type receptor do? C-type of receptor, it is, we have already mentioned, it is present on the surface, so it is plasma membrane receptor. And C-type of receptor, it is antifungal in nature. Fungal in nature. That's all. Now let's move toward node-like. Okay, before moving toward node-like, let's move toward rig-like receptor. What is rig-like receptor? Rig-like receptor functions as interferon gamma. That means it is also antiviral in nature. Okay, now we need to know about node-like receptor, and we are going to talk a little bit more about node-like receptor. What is node-like receptor? Okay, talking about node-like receptor identifies bacteria. It recognizes bacteria. It recognizes low potassium content inside the cytoplasm. It also recognizes reactive oxygen species. It also recognizes uric acid. And one important point that we need to know while talking about node-like receptor is it performs functions by producing inflammasome. Okay, now what is this inflammasome? Okay, before getting into inflammasome, I would like to take you toward picture from 10th edition Robbins, and this will make clear. Okay, the same thing is going on here. These are the extracellular or the plasma membrane receptors that are present outside the membrane receptors, and the inside these are the cytosolic. Okay, we haven't mentioned about endosomal. We have talked about this one, extracellular and cytosolic only right now. So as we have mentioned earlier, this is toll-like receptor that we have already mentioned, and this is lectin, C-type lectin receptor. C-type lectin receptor, I have already mentioned toll-like receptor. What it does, it receives antigen. Look here, we can see here, it is a bacterial product antigen. It receives bacterial product and then it releases what? Nucleus factor K beta and another interferon gamma. The same thing again, I'm repeating it. What does C-type of receptor do? It receives microbial polysaccharide or antigen, and then it performs what? Antifungal function. That's all. This was the thing that we were talking about, the one receptor present outside the cell, in the surface of cell, we can say. And the cytosolic, we learned already about node-like receptor that helps to identify bacteria, and also the product of damaged cells, we can say uric acid, ROS, we already said about it. And rig-like receptor, what is rig-like receptor? These produce interferon gamma, and we have already talked about it. So this was a brief introduction about the pattern recognition receptors. Okay, now talking about node-like receptor. What we're talking about node-like receptor, we just paused our conversation in talking about while talking about inflammation. Now what is inflammation? Okay, before getting into inflammation, we need to know about the node-like receptor. What is node-like receptor? Exactly, while talking about node-like receptor, you can see this is the cell membrane, and inside we are talking all about the cytoplasmic, because the node-like receptor that you can see right here is cytoplasmic membrane. This is node-like receptor, and here you can see it is node-like receptor and P is written, and it is written P, it is written at the end there is P because it contains domain pyridine. It contains domain pyridine. Okay, now what I said was I just brought one word, and the word was inflammation. INFLAMMASOME. Okay, there is one thing that you need to know about inflammation. What is inflammation? Okay, it is the protein. I mean, it is a protein that is produced in combination of node-like receptor, adapter, and caspase 1, which is in inactive form. Okay, before getting into that thing, I would like to tell that activation node-like receptor activation is associated with what? Activation of inflammation or production of inflammation, and this inflammation will cause activation of caspase 1, which releases interleukin 1 that is responsible for causing fever. Okay, you might not get everything that I said right now, but okay, now we'll get into it. Okay, this was what? Node-like receptor. This was node-like receptor, and this is adapter that you can see right here. This is adapter, and here you can see caspase 1, which is in inactive form. Okay, when these node-like receptor combined with adapter, you can see here, this was node-like receptor. As you can see here, this is node-like receptor. This node-like receptor is combining with what? This adapter that you can see here, this adapter. Okay, node-like receptor combined with adapter. When these two are combined or these two are attached, then after we can see that caspase 1, which is in inactive form, also comes and attach here. Okay, everything, whatever it is shown here, these forms inflammation, or talking particularly, we can say node-like receptor with domain pyrin inflammation. This is what inflammation means. Now, what is this inflammation causing? This inflammation, which was having what? Caspase 1 in inactive form, adapter, and then node-like receptor, receptor with domain pyrin 3. These, after formation of this inflammation, it will cause caspase 1 to detach from it, and this now, before attaching it was in inactive form, but after attaching it came into active form. That means caspase 1 is now in active form. It will be active as soon as detached from here. So the caspase 1 now is in active form, and this caspase 1, what it does is it produces interleukin 1. And what this interleukin 1 does? This interleukin 1 plays significant role in inflammation and also cause fever. This is the same thing, fever. Now I think you might be clear about node-like receptor inflammation. Okay, we can just write here one simple straight line, and the line says is inflammation. What inflammation does? Inflammation does is it activate, okay, caspase 1. It activates caspase 1. What caspase 1 does is it releases interleukin 1. Interleukin 1 release, and what interleukin 1 does is it causes fever and plays important role in inflammation. Okay, inflammation, I wrote it like this. So that was all about node-like receptor and these and that things. So still till now we were talking about what? We were talking about different receptors, right? We're talking about different receptors. Right now we just completed node-like receptor. Now after this we will just move toward a few words about adaptive immunity, because right now we were talking about innate immunity. Completely we were talking about innate immunity. Now let's focus about adaptive immunity. Okay, talking about adaptive immunity, talking about adaptive immunity, simply we said it is acquired during the lifetime whenever antigen enters in our body. So we said it can be called as acquired immunity. So what are the things that, or we can say what are the component of acquired immunity? We already said the component of innate immunity, that were what? That were different cells, the barriers, right? Plasma proteins. We talked about different receptors. Now in adaptive immunity we have two components, and the two components are, one is B cell and another is T cell. Okay, talking about B cell and T cell, at first let's understand the origin. What is the origin? We know in hematopoiesis, okay, I just wrote here hematopoiesis. Okay, well, dealing with hematopoiesis we have initially what? hematopoietic stem cells. We have hematopoietic stem cells. Okay, let me write it like this: hematopoietic stem cells. Hematopoietic stem cells will looking toward the timeline of this our B cell and T cells. We got what? We have, okay, let's only draw only one thing. We have lymphoblast. What does this lymphoblast does? It differentiate into B cell, T cell, and then natural killer cell. Okay, what are these things now? B cells. Okay, we have already talked about B cells and T cells. These are the component of what? Adaptive immunity. And natural killer cells, they are the component of innate immunity. Okay, now B cell and T cell, what happens is when lymphoblast is formed, some of the lymphoblast goes to thymus and they mature there and form T cells, and some of the lymphoblasts, they just remain in bone marrow, and while they remain in bone marrow, they form B cells. And some of the lymphoblasts, they don't change. When they don't change, they are called knife cells. Okay, these don't change. They don't change. They have the similar function as of lymphoblast. They don't change, and they are called the knife cells. Okay, this was the origin. Now, where we're talking about B cells and T cells. Okay, talking about B cells and T cells, I already mentioned B cells get maturation at bone marrow and T cells get maturation as thymus. Okay, now just thoroughly I will say about B cells and T cells that we need to know right now, because in detail we are going to learn about B cells and T cell in separate lecture. Okay, B cells. Whenever we activate B cells, at that time what happens? B cells converts into plasma cells. Now what this plasma cell does? This plasma cell secretes antibody, and we can say this antibody immunoglobulin. And these antibody are mainly present in fluid of body. So the action of B cell, the action by which B cell provides immunity is also called humoral immunity. Okay, the word humoral means what? Fluid. That means B cell, what B cell does? The B cell performs humoral immunity or plays role in providing humoral immunity. Now talking about T cell, what T cell does is T cells provide what? Cellular immunity. Now we'll never confuse about humoral and cellular immunity. B cells performs humoral immunity, whereas T cells perform cellular immunity. Okay, and another important thing is humoral immunity is against extracellular microbes. What I said, humoral immunity is effective against extra microsomal microbes, or we can say simply extracellular microbes. That means the microorganisms that are present outside, they are acted by what? Humoral immunity, or we can say they are acted by what? B cell. And simply we can remember it like this: B cell, B means bahira, bahira means what? Outside, extracellular. We can say it like that. And similarly T cells are effective against what? They are effective against intracellular microbes. Okay, these are against intracellular microbes. Okay, we have talked. Okay, what we did today is we simply talked about, we talked about difference between innate immunity and adaptive immunity. We talked about their basic differences. Okay, now here is figure, we can see it now. Okay, here we can see the component of innate immunity, epithelial barrier. these the yellow things that are right here are microbes. These are microbes, and these microbes are prevented by epithelial barriers to enter in our body. Similarly, these are the phagocytes, and under phagocytes we talked about what? Neutrophils and macrophages, and these are dendritic cells. And dendritic cells, we can dendritic cells especially, they are present in skin and also in spleen and lymph nodes, and the one that are present on skin, they are called what? Langerhans cells. And the one that are present in lymph node and spleen, they are called follicular cells. We'll talk that in detail in dendritic cell topic. And other component of innate immunity are complements, NK cells. Similarly, with adaptive immunity, we said the example of adaptive immunity are the component of adaptive immunity are B lymphocyte. Okay, we can see here B lymphocyte, it is the B lymphocyte, and this is microbe, and this B lymphocyte is performing action against these microbes by producing antibodies. You can see here these are the antibodies. Okay, and we can see here T lymphocyte also. We'll learn these things in detail, don't worry. T lymphocytes, the similar case we can see here. So this was all about immunity and the introduction to immunity. And the next topic that we'll be talking about is B cells. What I said is, now we'll be talking about B cells. What are these B cells? B cells are the component of what? Innate or adaptive immunity. These are the component of, okay, it's not working like this, let me do it. Okay, that's fine. B cells are the component of adaptive immunity, right, which was acquired during the lifetime when microorganisms were entering in our body. Okay, initially we're talking about B cells. The thing that we need to know is we need to know about their locations where they are present. Okay, B cells, I hope you remember in last lecture we were talking about hematopoiesis, in which we talked about B cells, T cells and natural killer cells, how and where they are formed. So here we can see B cells. Talking about the location of B cells, I would like to mention the B cells are present on lymph node, spleen, GIT, tonsils. Talking about these things, we need to learn so well about lymph node. I would like you to focus on this amazing figure of lymph node. This and then this. Okay, I think it's not symmetric, but it will work. Okay, inside, this is the lymph node. Inside the lymph node, I mean in this area, okay, this outer one is cortex. This is cortex, and inner to cortex we have paracortex, and inside a paracortex we have medulla. Okay, we said the B cells are found on lymph node, but where exactly in lymph node? So we're talking about lymph node. The exact position in lymph node where B cells are found is cortex. That means in cortex we'll find B cells. Similarly, in paracortex we'll find T cells. Okay, lymph node has got room for all. And in medulla we have got macrophages. Okay, this is so important, we need to just memorize it, or it works similarly. We can say B means bahira, right? Bahira means cortex, or we can say it is the place for B cells. This was all about lymph node. In lymph node we're talking about GIT. Okay, let's talk about GIT. We're talking about GIT. In GIT, where we find B cells? We find B cells in ileum. ILEUM. Okay, ileum is a large structure. Where exactly in ileum? We're talking about exact position inside ileum. There is present Pierce patches. Okay, we find it in Pierce patches. Okay, where exactly in Pierce patches? We find in MALT. What are these MALT? This MALT is mucosa associated lymphoid tissue. What I said is mucosa associated lymphoid tissue. Okay, that means B cells in GIT are found in mucosa associated lymphoid tissue, MALT, or these can be also called as mucosal tissue. This is also called as mucosal tissue. It's the same thing, mucosal tissue. Okay, why we are talking about this thing right now? We are talking about this thing because whenever there will be ulceration in Pierce patches, ulceration present in Pierce patches, at that time which cell will be the most effective affected cell? Which cell will be the most affected cell? At the time we will say it is the B cells. Okay, Pierce patches, destruction in Pierce patches or ulceration in Pierce patches means what? Ulceration in what? MALT, mucosa associated lymphoid tissue, and this means there is the presence of what? A large amount of B cells. That means what will be affected? B cells will be affected. So it was a clinical correlations or microbiological correlations. Okay, talking about B cells, we need to know what are the chemical marker of B cells. What does chemical marker means? Chemical marker means what makes B cell a big B cell. I means how will identify B cell? By looking toward its marker. Okay, the thing that we are talking about right now is we are talking about B cell markers. What I said is let me zoom it out, B cell markers. Okay, B cells marker. Talking about B cell marker, we need to know, okay, I'm gonna mention the B cell markers, and they are CD molecules, CD 19, 20, 21, 22 and 23. These are, what are these? These are B cells marker, right? B cells marker. These are B cells marker. Okay, another, okay, I'm gonna mention it initially and then I will describe the important thing that we need to know from these markers. And we need to mention CD40 also. And another thing that we need to mention is CD19. Other thing that we need to mention is CD20. We have already mentioned it above, but again we are writing it down because we need to know a bit about them. These are the B cells marker. Simply, the B cells marker are the certain marker, certain things that are present on the surface of B cells that will help us to identify the B cell. Okay, that's all. Okay, we're talking about 19, 20, 21 and 22, 23. The important point about CD21 is the receptor. Okay, let me write. It acts as receptor for Epstein-Barr virus. That is the important relation about it. And we're talking about 19. The 19 one, or here one, the 19, CD19 is the marker that is always expressed in surface of B cell at any maturation, at any stage of maturation. That means it is expressed. CD19 is expressed on surface of what? Surface of B cell at any time of its maturation. So such type of marker which are present throughout the lifetime of B cell, they are known as pan B cells or pan B markers. Okay, what I said here is they are called as pan B cell markers. Similarly, similarly as it has CD19, there is also a specific CD molecule that is present on the surface of T cells throughout its life, and that is called what? Pan T cell marker. It's similar. Okay, and we are talking about CD19. Okay, now let's talk about CD40. Okay, what is this CD40? CD40, okay, I'm gonna write it here, CD40. Okay, let's erase it. We're talking about CD40 is also a marker obviously of B cells, and the important point about CD40 is it attaches with CD40 ligand, and this CD40 ligand is present in T cell, and that is the important relations between T cell and B cell. That means who connects these two cells? The one that is connecting these two cells are CD14 in B cells and CD40 ligand in T cells. That is the important thing about it. And then another thing that we need to know is, and it is for CD40 only, and the CD40 and CD40 ligand, they help in switching between the antibodies. Okay, that will be clear after some time, and we'll know about these things when I will draw a brief diagram about B cells and how it is interacting with T cells. So just remain here. And another one that I'm going to mention is CD80. Okay, just about CD40 and CD40. Okay, I'm going to write few points, just mention it here, and later on we can revise it again. Okay, the thing is these two bind each other. That is one point that we have already mentioned. And another thing is these two are responsible for B and T cell interaction that we have already mentioned. And the next point is that we need to know is this help in quality antibody formation. What I said is quality antibody formation. This is what we will be talking about, antibody switching, later on. So let it go. Now CD80 and CD86. Okay, these two molecules interact with CD28. They interact with CD28, and this interaction with CD28 will help in signaling. And what is that signaling? Co-stimulatory signaling. Okay, what is this co-stimulatory signaling? We'll know later on. Don't worry, we'll know later on. We'll be clear by the end of this lecture. Okay, we're mentioning about how B cell, or how B cell produce antibody is. B cell, we got B cell, and we're talking about B cells. When B cells are activated, right, they form what? They form plasma cells. And when these plasma cells secretes antibody or immunoglobulin. That is what it is. Now we talked about B cells. Now we need to talk about this antibody or immunoglobulin. Okay, I'm going to draw a basic structure right now. I'm going to draw a basic structure that you need to be clear. Now I think, okay, just look it out, just watch it. The basic structure that I'm going to draw of antibody is, we got here, and this is Y-shaped, antibody is Y-shaped. I need you to focus it right here. This is what we call Hypogen. What I wrote here is Hypogen. And now these, okay, let me try it again. These are light chain, and two Hypogen are joined to each other by what? Disulfide bond. That is all. disulfide bond. And we can see disulfide bond not only there, we can see it here too. And in this antibody we can find here one region, and that region is FAB. What is that region called? FAB. And in this FAB region there is the attachment of antigen. Okay, now let's make ourselves clear. There was B cell that was producing antibody, and what this antibody was doing, this antibody is attacking or is attaching antigen with it. This is how it works. Okay, now as I have already drawn a basic diagram about antibody, I would like you to take toward. Okay, one thing is clear, that antibody and immunoglobulin are the same thing. Don't confuse between them. And there is light chain in it, right? And this light chain are called what? Kappa and sigma, and another heavy chain. Talking about heavy chain, these heavy chains are of how many types? 5 types. These were of 2 types. These heavy chains are of 5 types. What are these? Okay, alpha, sigma, delta, epsilon, naught. These are of 5 types. Okay, we're talking about antibody. This antibody or immunoglobulin are classified into how many types? This is classified into how many types? GAMDE, 5 types. These are classified into 5 types. These are IgG, IgA, IgM, IgD and IgE. These are the classification of antibody or immunoglobulin. And this classification is based on what? This classification is based on heavy chain. This is one of the important MCQ. Okay, I already said we're writing it in order of GAMDE. Why this? Because G is having maximum concentration, and IgE is having minimum concentration. Okay, now serially we'll learn about each antigens, and we're learning about each antigens. Let's begin our discussion with IgG. Okay, first point I've already mentioned that IgG has what? It has maximum concentration in body. I already mentioned about it, maximum concentration in body. Similarly, other thing that we need to know about IgG is, just write C and C. That's, we can write here in concentration also, we can write here C, maximum concentration. It's all right. Okay, this C, first C is it crosses placenta. Another C is it activates complement system or complement activation. Another C is it makes cells tastier. And what we call the term cell making cell tastier, opsonization. Okay, this was about IgG. Okay, now GA. Now we need to talk about IgA. While talking about IgA, okay, the first thing that we need to know about IgA is it mainly exists in two forms. What are the 2 forms? The 2 forms are, one is monomer, simply only one molecule. Monomer means made up of only one molecule, and the monomer form is found in serum, right? And another form that we'll be talking about is it is found in dimer form. And this dimer form of IgA is found in secretions. secretions and mucosa. Okay, we say dimer form, but how exactly? I mean, dimer form means simply what? Dimer form means simply this is one and this is another. Okay, let's make light chain also, and these two are connected to each other by what? These are connected by J. What is this? J. We call it J. What? J. J chain. IgA exists in dimer form in secretion and mucosa, and dimer form. In dimer form, two IgA antibody are connected to each other by what? J chain. That is the important point that we need to know. And another thing that we need to know here is, just highlight A here and just write A. Okay, A means what? The first letter of alphabet, and similarly it is the first line of defense. That means whenever antigen attack our body, the first antigen, the first antibody that needs to be produced or that will defend us is what? IgA. It is what? First line of defense. First line of defense. And again write another A, and this another A means it activates alternate pathway of complement system. Obviously, activation of alternate pathway of complement system. Yeah, we have already known there are two pathway. One is classical pathway and another is alternate pathway. It activates alternate pathway of complement system. Okay, complement system, not complement pathway. Okay, this was all about IgA. We have talked about two immunoglobulins right now. Now let's move into another one, and another one is, now we'll be talking about what? We'll be talking GAM. That means IgM. Talking about IgM, the thing that we need to know is IgM. Just go through word M. It has what? Maximum size. Go with same flow that I'm going. Okay, it has maximum size. I'm talking about size, maximum concentration. It is of IgG. Maximum size. It has got maximum size, so it has got what? Maximum molecular weight. Okay, maximum molecular weight. It has got maximum molecular weight because it is in pentameric form. What I said, pentameric form. Pentameric form means there are 5 immunoglobulins that will attach together and form IgM. Obviously, we can say it is in pentameric form since it is in pentameric form. Okay, since it is in pentameric form, that means many. It has many. Penta means 5. 5 means many. It is also called millionaire antibody. What I said is millionaire. Antibody. Okay, so these 4 points can be related to each other. I mean, we can just memorize them by relating with one another. And the next point that we need to know about IgM is, okay, the next thing that we need to know about IgM is, this is the component of primary immune response. Okay, what I said, component of primary immune response. Okay, I think before this we said IgA as first line defense. Now we are telling this is the component of primary immune response. What does that mean? Okay, it is the component of primary immune response means, okay, when antigen has already entered in our body, it will act, that antigen will activate immune system, and that immune system will activate a lot of antibody, and that first antibody which is produced by activation of immune system is IgM. and that first response is called primary response, primary immune response. So you will be clear when I will be talking about how antigen enters in the body and what B cell does and what is the relation of T cells, and we will be covering this topic in, I think, basic about immune reaction. At that time we will talk about these things. At that time we will be more clear about this thing. So component of primary immune response, it is a component of primary immune response. So, and another thing is, if, okay, we said that it is in pentameric form, but if there will be presence of IgM in monomeric form, then it will form as B cell receptor. That means in pentameric form it has all these functions, but in monomeric form it acts as B cell receptor. Okay, B cell receptor. Okay, let's keep it simple, this much only about IgM. Now let's go toward, okay, where we are, GAMD, IgD. Okay, there is not much to tell about IgD, because simply the thing that we are going to write is acts as B cell receptor. That's all. And B cell receptor, we just mentioned that monomeric form of IgM also acts as B cell receptor. So B cells receptor, what are two things that acts as B cell receptor? That are IgM and IgD. That is all. Now talking about IgE, I think the last one, yeah, it is the last one, IgE. The first thing that we know about this last one is, obviously, it has what? It has minimum serum concentration. We have already mentioned about this thing. Okay, and another important thing is this IgE. These are attached on the surface of mast cells. These are attached on surface of mast cell. And what this results? This results hypersensitivity 1 reaction. Okay, hypersensitivity reaction 1. And this is the heat level antibody. Heat level antibody. Okay, this was all about what? This was all about our 5 antibodies, and these 5 antibodies are based on the basis of Hypogen, and this was all about the immunoglobulins or the antibodies. Now the further we will learn about, okay, the relation till now. Let's draw a map. The map is, we got here antibody, we got here antigen. This antigen was detected by antibody, right? Antibody. Till now we have learned briefly about antibody. Now we'll learn about antigen. What we'll be learning about is antigen. We're learning about antigen. We'll learn about 2 types of antigen. Simply, antigens are microorganism, viruses, bacteria that try to invade in our body, that try to get entered in our body. These are the antigens. Okay, talking about antigens, there are 2 types of antigens. Okay, the first type of antigen that we'll be talking about is non-proteinaceous antigen. What I wrote? Non-proteinaceous. Non-proteinaceous antigen. Now what are these non-proteinaceous antigen? The antigen that does not require additional support of cells. That means there is one antigen that is flowing in my circulation. I mean free antigen. There is free antigen. That free antigen is just revolving in my circulation. At that time, that free antigen is captured by my B cells. If the B cell which is capturing that antigen will not require any other additional support from T cell or any of the cells, then the antigen that it is capturing is non-proteinaceous antigen. Okay, what I said is, let's suppose this is our B cell. This is our B cell, and on the surface, now the thing will get clear when I start drawing. On the surface we have B cell receptor. Now this B cell receptor will capture one antigen. Okay, this is antigen. Okay, let's not draw with the same color. This is the antigen. This is what? This is antigen. Now without help of any other cells, if this B cell, okay, let me write with green, this B cell will produce antibody, and that specific antibody which produced in primary immune response is IgM, we have already mentioned. If it produces antibody without the help of any other cells, then this antigen is called non-proteinaceous antigen. That's all. What I said is, do not require support of any additional cells. That's all. That is the one point. And another thing is, since I've already mentioned that it does not require support of any additional cells, that meant it does not require support of T cells also, right? That's all. And this non-proteinaceous cells produces what? Antibody. And what is that antibody? The antibody is IgM, the antibody of prime immune response. That's all. That is non-proteinaceous antigen. Okay, now talking about another one. What is the another one? The another one is proteinaceous antigen. Proteinaceous, proteinaceous antigen. What is this proteinaceous antigens? The another name for proteinaceous antigen is T cell dependent antigen. T cell dependent antigen. The point are clear from this second name only. That means this antigen will be attached to what? This will be attached to B cell, but that B cell will itself not be able to produce antibody. It will require the help of what? T cell. Here comes the point. T cells dependent. That means requires the support of T cells for the proper activation of immune system. That's all. That means now we ended up our discussion about B cell. Now we are going to enter into what? T cell. Now let's jump into T cells directly. I would like to change the page. The discussions about T cell. Okay. Talking about T cells. What are T cells? We have already that map in our mind of hematopoietic stem cell, how it gives T cells, and what are T cells. Okay, we have already that thing in our mind, so we'll not dwell there and we'll just proceed forward. Okay, talking about T cells. What is the site of T cells? What are the site? Where are T cells found? T cells are found in, they gain their maturation in thymus, so the name T cell is given. That means thymus. They get maturations. Now the site of T cells, that means where are they present? From previous lecture we have already known the B in lymph node. In outer region, in cortex, we had B cells. Similarly, in paracortex we had what? We had T cells. So in lymph node what? In paracortex. Similarly, in spleen we have T cell in PALS. What I said? PALS. What is this PALS? This is periarteriolar lymphoid sheath. Just focus on the word periarteriolar lymphoid sheath. Okay, that means in outer region of arteries there is lymphoid sheath, and at that site we can find what? T cell in spleen. That's all. Periarteriolar lymphoid sheath. Okay, similarly another location that we need to know where T cell are found is in GIT. In GIT, where we find them? We find them inside epithelial lining, or we can say intraepithelial lymphocytes. Lymphocytes. We can say it as IELS. Okay, in GIT, where we are finding B cells, we are finding B cells in Pierce patches. And where exactly in Pierce patches? We are finding Pierce patches exactly in what? MALTs, mucosal associated lymphoid tissues. That's all. And in GIT, we are finding T cells inside of epithelium lymphocytes, and that we can say intraepithelial lymphocytes. That's all. That were the site of T cells. Now, okay, let me draw one T cell. Okay, let us suppose, okay, let me make it a bit thicker. Let us suppose this is T cell. The T cell has, in the center, it has got nucleus. Okay, in the periphery, it has got CD molecules. What are these CD molecules? We have already mentioned some CD molecules in B cells, but we are not talking, we are not concerned about what these CD molecules were. Okay, these are what called cluster differentiating molecules. These are CD. Okay, let me write here. CD molecules are what? Cluster differentiating molecules. What does this mean? Cluster differentiating molecules means what? They will differentiate T cells on the basis of what type of CD cells are present on the surface. Okay, that is what it is. And another thing that T cell contains is it contains T cell receptor. What I said? I said T cell receptor. This is T cell receptor. The two things, one is the CD cluster differentiating molecules, and another thing that we're talking here is TCR, T cell receptors. What this T cell receptor does? Okay, these T cell receptors, they receives antigen. Okay, let me just make APC. What is APC? We haven't talked about APC yet, but we will be talking about APC in further topic. Okay, this is APC. What is this APC? This is antigen presenting cell. That means it presents antigen. From name it is clear that it presents antigen. Okay, that means this in APC we have MHC molecule, and this MHC molecule will present what? This MHC molecule will present these antigens. What is this? This is antigen. This MHC molecule will present antigen to what? T cell receptor. That is the function of T cell receptor. Okay, this is antigen presenting cell. This is what? This is antigen. It's not so clear. No, okay, that's fine. This is antigen. This was antigen presenting cells. So what happens here? Okay, T cell. This is T cell. The first thing that we need to know is before in B cells we learned about non-proteinaceous antigen. The non-proteinaceous antigen were what? They did not require T cells to activate. I mean, they did not require any other cell to produce antibody. But this T cell will only receive those antigen which are not in free state. That means the antigen that are processed from antigen presenting cell or that are processed from any other B cells will only be detected by T cells. Okay, I'm going to tell one statement here. T cells get activated by pre-bound antigens. These antigens is pre-bound to what? APC. T cell never get activated by free antigens or the non-proteinaceous antigen that we mentioned earlier, where we mentioned here non-proteinaceous antigen. So these are not activated by non-proteinaceous antigen. Okay, that's all. Now what is the next thing that we need to know? The next thing that we need to know is CD molecules. What do we say? These CD molecules are present on the surface of what? On the surface of T cells. What are their importance? Their importance is they helps in differentiating. Simply, CD molecules are useful in identifying the certain cells. That's all. Okay. Okay, now we are going to mention CD molecules. Okay. CD molecules, we have got CD1, we have got 2, 3, 4, 5, 6, 7 and 8, and these are the T cell markers. What are the markers? Markers are specific molecules that are present on specific cells by which we can identify that particular cells, right? Okay, I'm T cell marker because why I'm T cell marker? Because I have CD1 to CD8. That's why I'm T cell marker. Okay, sorry, that's why I'm T cell. That's all. Now another CD molecule that we need to mention is about CD28. What is this CD28? CD28 is CD molecule on the surface of T cell, obviously, and what it does is it binds with CD molecules on B cells. If you remember last time what we studied is CD86 and 80. Okay, let me show you. Last time we did is here CD80 and 86 binds with CD28. What this CD20 where this CD28 was? It was on T cell. So now we have got here our discussion in which we are talking about CD28. What is this CD28? CD28 is CD molecule on the surface of T cell that interact with what? CD80. Okay, let me do it. That interacts with CD80 or CD86, and through this interaction it was providing one signaling. And what was that signaling? That was co-stimulatory. What? Co-stimulatory signaling. Co-stimulatory co-stimulatory signaling. Okay, this was all. Okay, we talked about CD1 to CD8, CD28, right? These CD80 and CD86, they are CD molecules of what? B cells. Don't forget about these things. That means the B cell and T cell are interacting with each other. That's all. Now talking about another important, this CD3 molecule. CD3 molecules is pan T cell marker. Okay, what I said? Pan T cell marker. Okay, in B cell also I have mentioned that pan B cell marker was what? It was CD19, and here the pan T cell marker is CD3. What is pan T cell marker? Pan T cell marker is in any of maturation phase of T cell there is presence of one CD molecule, and that one CD molecule is CD3. That is what pan T cell marker is all about. Okay, now CD molecule, it is pan T cell marker. Okay, another important thing that you need to know about CD3 molecule is it helps in signal transduction. SIGNAL, signal transduction. Okay, signal transduction. Okay, signal transduction. The molecule that you see right here just below T cell receptor, this molecule is CD3 molecule. That means whenever TCR, T cell receptor, receives antigen, then it will pass information to what? CD3 molecule, and this CD molecule will pass signal to what? Nucleus. Right here in center we have nucleus, and this nucleus will cause T cell activation. That is what it is. So CD3 molecule, signal transduction and pan T cell marker. Another thing that we need to know is this causes T cell activation. That means CD3 molecule will just send signal to nucleus, and then that will cause activation of T cells. There are other component that helps in T cell activation also. And what are that other component? They are calciurenin and interleukin 2. That means activation, T cells activation is also done by, assisted by calciurenin, calcineurin and interleukin 2. That's all. Okay, now what I said is, there is antigen presenting cell. Antigen is presented because free antigens are not pre-antigens cannot activate T cells. So antigen which is processed by APC, antigen presenting cells, are received by T cell receptor. Antigen received by T cell receptor here, and this T cell receptor receptors have CD3 molecules on its base, and the CD3 molecule will send signal to nucleus, and this nucleus will tell this T cell to activate, and that is how the T cell is activated. Now talking about T cell, we have got two type of T cells. Okay, there are two type of T cells. What are the two type of T cells? The two type of T cells are, number one CD4 T cell, CD4 T cells, and another one is CD8 T cell. And on what basis we are classifying these T cells? We are classifying these T cells on the basis of CD molecules present on the surface of T cells. If in surface there is CD4 T cells, then obviously that is CD4 T cells, and if in the surface there is CD8 molecule present, then that is what? That is CD8 T cells. That's all. And another name for CD4 T cell is, it is also called the helper T cells. What? Helper. And CD8 molecules, another name is cytotoxic. Okay. CD4 T cells, it is further classified into subtypes. What are the subtypes? The further subtypes are helper T cell subtype 1. This is helper T cell subtype 1. Another helper T cell subtype 2, helper T cell subtype 17. Now what are the function of helper T cell subtype 1, 2 and 17? 1 is responsible for type 4 hypertensive reaction, and it is also responsible for secretion secretion of interleukin 2 and interferon gamma. Okay, what about helper T cell subtype 2? Helper T cell subtype 2 help in, or it is responsible for performing hypertensive reaction 1, and it also releases or secretes interleukin 4 and 5. And talking about helper T cell 17, helper T subtype cell 17, it secretes interleukin 17.